Parasim, and in my second video I'll be explaining about the citric acid cycle. Citric acid cycle is also known as the tricarboxylic acid cycle, TCA, which is a biochemical pathway used to generate energy through oxidation of acetyl-CoA. It is also used as synthesis of NADH and the production of the amino acids. Citric acid cycle takes place in mitochondria of eukaryotes and the cytosol of prokaryotes, and it also occurs in the matrix of the mitochondria. First step involved is actually a preparatory steps which begins with pyruvate. It is derived through glycolysis of glucose, which is a 6-carbon compound, and then split into 2 molecules of pyruvate, which is a 3-carbon compound. So the next step will be the oxidation of pyruvate in the acetyl-CoA by the enzyme called pyruvate dehydrogenase. In this reaction, a molecule of carbon dioxide and NADH is generated. The acetyl-CoA is a 2-carbon compound. So the first step will be the acetyl-CoA combines with the oxaloacetate, which is a 4-carbon compound, to form citric, and the resulting molecule is a 6-carbon compound. This reaction is catalyzed by the enzyme called citrate synthase. Now, second step will be the citrate is isomerized in isocitrate by the enzyme called aconitase. Now, a water molecule is removed and the other one is added to generate a structural isomer of citrate called isocitrate. In my third step, the isocitrate is oxidized into alpha ketoglutarate by the enzyme isocitrate dehydrogenase. In this reaction, a molecule of NAD is reduced to NADH, and there is also a removal of carbon dioxide, which is also known as the decarboxylation process. The alpha ketoglutarate is a 5-carbon compound since 1 carbon is lost into the carbon dioxide. In my fourth step, the alpha ketoglutarate is converted into succinyl-CoA by the enzyme called alpha ketoglutarate dehydrogenase. In this reaction, NAD is reduced to NADH and a molecule of carbon dioxide is released. Hence, succinyl-CoA is a 4-carbon compound. In my fifth step is about this succinyl-CoA is converted to succinate by the enzyme called succinyl-CoA synthetase. A molecule of GTP is generated, which here means it uses 1-phosphate to produce. There is a substrate level phosphorylation going on where there's a conversion of ADP to ADP. The same goes to GDP to GTP by the adding of the inorganic phosphate. In my sixth step, the succinate is converted into the fumarate by the enzyme called succinate dehydrogenase. A molecule of QH2 is generated, which is used for the production of FADH2. Basically, FAD is reduced to FADH2. Fumarate is converted to malate by the enzyme fumarase. 1 molecule of H2 is gained. Lastly, malate is converted to oxalate by the enzyme malate dehydrogenase, and NAD is reduced to NADH. So for every acetyl-CoA entering the cycle, it will produce 3 NADH, 1 FADH2, 1 ATP, and 2 carbon dioxide. Since glucose is split into 2 pyruvate compounds for each molecule of glucose, this cycle runs twice or maybe runs again. So the products formed are 6 NADH, 2 FADH, 2 ADP, and 4 carbon dioxide. So all NADH and FADH are next fed to the electron transport chain.